Okay, so good morning, everybody. Thank you very much for coming to this talk. So I've said I was going to introduce myself. I'm actually Claire Gachon. I've been here at SAMS for more time I care to remember. Really what I do is diseases of algae. So basically, I just wanted to very quickly here acknowledge all the funders that have funded my work in the recent years. And I've made a choice this time to actually not try to cover the depth of or the variety of projects that we are doing in my group, but really to just focus specifically on one that is a European funded project. It's called GENIALG, and it has to do with genetics, GWAS, and so forth. So I had decided to keep things short and simple, but then I decided that no, I just cannot do this. So I will talk about GENIALG at some point, but I will build up a bit of a picture first, talking about why diseases are important in algal aquaculture and really trying to bring some facts about that. So this is really algal aquaculture being a business, so really I would say the applied side of the science we do. Then I'm going to talk about the work we're doing on pathogens in this aquaculture setup. Then how we are moving a bit from doing science to talking to politicians, and I know I have to improve my picture of a politician here because it's not very flattering. Trying to look really into biosecurity and perhaps influencing legislation about biosecurity. Then I'm really going to move from, I would say, this global perspective to really narrowing down to what is going on at the moment in Europe, where effectively people are very interested into cultivating seaweed, and amongst all seaweed, Saccharina latissima, so kelp, and effectively how, as part of GENIALG, we are trying to put together the tools to breed for kelp. As a disclaimer, we are not breeding kelp yet. We're really just trying to set the scene, and that's really what I'm going to try to show. So yes, diseases as a threat to the algal aquaculture. So I think you've all seen this, which is really the sort of exponential growth of algal cultivation. My graph is a bit old, but it's really still going up every year, very quickly at 8 or 10% typically per year, and that's both in quantity and in value. So really that's now a billion-dollar business. Dominated by China, of course, but there is a lot of countries all across the 5 continents that are actually getting into this business. If I focus on laver, which is the sushi wrap type of alga, it's one of the most valuable industry in Asia. So this is what you've heard, okay? This is what you've seen about this industry. It's like sushi is good for you, it's very healthy, and so forth. The true story, however, is really the same as you will have in all agriculture and aquaculture production, is that you have a massive proportion of your crop that is lost to disease year on year. And it turns out that the 2 of the most important pathogens in the Asian industry are oomycetes, with nice names like Opidiopsis porphyri or Pythium porphyri. And they're my favorite organisms. So really, what is going on at the moment, and this is these figures are based on a study by Kwang-Hoon Kim, with whom I've been working a lot, is that about roughly 20 to 30% of the potential production is lost every year to diseases in that porphyra industry in Korea. But it's also the management measures that they're putting in place to try to avoid these diseases amounts to up to 50% of the running cost of the farm. So it's a massive burden economically and in terms of management to actually reach that number, which already seems horrible that you're losing a third of your crop, but you're losing a third of your crop despite having put 50% of your running cost into trying to protect against disease. So that's just like really a graph that is in this paper trying to show the incidence of disease in the 2 biggest Korean farms. And this is on 2 consecutive years. So you can look at temperatures and so forth, but really what I want to get out of this graph is that you've got 3 big hitters that are called red rot, green spot, and Opidiopsis, okay? And at any point of time when you're going in the farm, you have a seasonality of pathogens, but at any point of time you roughly have, say, 20% of your crop is infected with any one of these pathogens, right? And really, the interesting thing is that these pathogens here, so Pythium, the red rot agent, has been now known to science since the 50s. But this Olpidiopsis here has been described by Guang Hun in 2016, and the green spot disease agent has been described as a virus in 2017. So literally, you discover the pathogen as the industry grows, and this is a billion-dollar industry, right? Okay, coming back to Olpidiopsis, as I was saying, is some of our favorite parasites. This is the baseline of molecular knowledge, 18S barcode sequence that we had in 2015 when we started with this global seaweed project. And basically what you're seeing is that this is one of the biggest hitter in a billion-dollar industry, and you have 5 18S sequences worldwide. 3 of them come from Japan, Korea, and China, which are the producing countries. And then you had 2, I would say, amateur enthusiastic phycologists who just worked on this randomly out of curiosity. So one guy, John West, in Madagascar, and one in the Adriatic, Kyle Fletcher. Okay, and they're all intracellular pathogen of reds. And this is really where we were just not even 5 years ago, right? So what have you seen? We just decided to go far away. So as far away as the back beach here, and we went down to the Atlantic Bridge, which is 20 miles away from here, I think, not even 20 miles. And then sure enough, we just took the first random red algae we found, and we've essentially started to find Olpidiopsis in them. And we started to describe new species, and that's just published in Journal of Applied Phycology. And these are this red algae that we looked at, Apophyra, and also Palmaria, which are prime targets for cultivation in the UK. So really, the parasites, when you start looking for them, they are here, and they are unknown to science. Okay, then Yassine likes to play with computers, right? So what he did here was effectively that because of the revolution of next generation sequencing, there is now a lot of data, sequencing data, that are publicly accessible in GenBank, where essentially people have sequenced in bulk, I would say, environmental DNA. So say a bucket of seawater, what DNA is in it, and so forth. So what Yassine was doing was essentially programming a custom script that actually was able to use the Olpidiopsis that we knew, so the ones that are in black or in red, so the ones that were in the databases before or the ones that we've described, to actually fish any DNA signature that was similar to it. And then you put this on a phylogenetic tree. And the bottom line here is that we have maybe half a dozen now Olpidiopsis species that are known molecularly, so for which we know that the 18S sequence corresponds to an intracellular pathogen of red algae. And we find that there are at least 20 other species waiting to be described, right? And then when we put this back on a map with the GPS data that come with the sequence data, you see here in red the sequences that we know are Olpidiopsis. So this is still Asia. This is Scotland now, where this is the back beach, effectively. And a few other points, the Adriatic, Madagascar. And you see that we're able to kind of say that pretty much everywhere anyone has sampled, there are some of these organisms around. And you have here Australia, corresponds to all these green things here. We don't have GPS data, we just know it's a coastal program for Australia. But Australia seems to have a lot of biodiversity for these organisms as well. Okay. So coming from there, I think really moved into increasingly applied science. So I think I see myself now as the person kind of trying to build the science that is needed by industry and by the politicians to actually help build a sustainable industry. So that's the sort of quest for sustainability that seems to be very fashionable and I would say important at the moment. Okay, so that's really starting to look at a worldwide perspective. And we've moved a bit away from the laver industry at the moment, and we are now looking at the euchymid cultivation, and that's happening via the Global Seaweed Star program. And that is mostly done in developing countries. So same thing, you've heard, and it's true, that euchymid cultivation has started and the 60s, late 60s, early 70s, and that it's been introduced in different countries. And for example, it has promoted women emancipation in a lot of places. Here you've got Tanzania. So you've got women that have become economically active, and it's all great and so forth. I cannot be suspected of not appreciating this. Obviously, you've all eaten this sort of flickering type of desserts. Okay, so whenever you eat something like that, probably some empowered woman has contributed to growing it somewhere. The true story, however, is that a lot of this production is also threatened by diseases. So it turns out that the two major diseases of euchemoids are collectively known as ice and epiphytes, typically red algal epiphytes that grew at the surface of the alga and sort of compromise the integrity of the algal surface, and then it's the beginning of the end with the alga rotting. And I'm not into catastrophism, but it's really, in some places, it has led to complete collapse of the industry. This is an extreme example that is taken from a paper from Flower Msuya that we're working with in Tanzania. And that's really where the production of one particular species, Caprophycus arvaricii, peaked at around 1,000 tons in the early 2000s, and then was struck by these two diseases and then really collapsed to pretty much nothing in 2008. And it's barely recovered ever since. And basically, people are just going out of that business, trying to cultivate other species that are much less rentable, more resistant to disease, but it's really affected the communities. Now, this is I apologize for the people who've seen this for a long time. It's been in prep for like the last 5 years, I think. But I was trying to look at a more global perspective of what is going on in this industry. So here you're looking at I would say the origin, the center of diversity of the euchemoids, and that's really the coral triangle where you've got Indonesia, Malaysia, and the Philippines, typically. And essentially, you can see that they were pioneering the introduction of cultivation in the early 70s. Then the first date you see is the first date of a disease report I could find. And it's basically within 5-10 years of the industry starting, you have someone at some point publishing something saying, hey, I've seen this thing in my farm. But don't worry too much. Typically, that's what they say. And then, so ice are in red, endophytes are in yellow. And then the second date is the date of the crash, right? So say you start cultivating in 1969. It takes 6 years for you to see the first ice incident in the Philippines. Then in the early 2000s, you crash. And from the moment it crashes, it never recovers. That's basically what it is. Or it's an ongoing issue. Right, and that's in the Philippines. And then from the Philippines, you've had a lot of introductions in all other places. And then Tanzania, which I was just showing, the cultivation started in 1990. First disease incidence reported in 1995, crash 2006. You've just seen the figures. It's not recovered since. So effectively, what I'm trying to get out of this is that when you introduce an industry somewhere, you have what I would call a grace period of basically everything is fine or fine-ish. And then after that, diseases typically worsen. That's typically due to the intensification and sometimes to the industrialization of the industry, right? And it's not always the case. I don't want to go into catastrophism, but really sometimes it can lead to complete collapse, regionally collapse of the industry with extreme consequences, I would say social consequences on the people who are engaged in that business, right? And really, that once you're out of your grace period and once diseases are declared, they just don't go away. So, you can't just hope that you've sorted the problem for this year and it won't be back. It will be back. Okay, so this is perhaps a bit more, how to say, it's less neutral, I would say. It's about the perception of the industry by people and the evolving perception of the industry, right? I'm just looking here at people who've been describing and reporting about the industry in Tanzania. And it's interesting to see that at the beginning you have private sector, of whom Eric Ask is really a representative, saying like in many islands and coastal areas, eucumbia has become the crop with the highest total export earnings. In some cases, this has occurred within less than 5 years of being introduced. So effectively, at the beginning, it's like every singing and dancing industry where private sector is very happy to communicate, and for good reason, right? Women have come out of poverty, they've sent kids to school and so forth. I'm not denying that social benefit. But you really have like this, everything is fine type of history. And then you start, you start having the alarm bells ringing in the early 90s, so before the collapse happens. Okay, so you've got a guy, scientist, saying a potential threat to eucumbia cultivation in Zanzibar is the narrow genetic base. And I'll come back to genetic uniformity. Okay, this is typical. The more you select, the more you get a uniform crop, and the more you're vulnerable to diseases coming in. Then you start having private sector suggesting that more research is needed, right? And then it's saying 2000, right? So that's maybe 5 years before the crash. Understanding and developing the capability to mitigate or eliminate pests, herbivore, and diseases need to be addressed more closely. And that is Mr. Ask from the private sector, who is on my next slide as well. And he says there is increasing concern over the impact of exotic species have on endemic ecosystem and biodiversity. So I'm not talking diseases here, I'm really talking about introduction of a robust, fast-growing alga in an ecosystem, typically where coral reefs were, and whereby, well, you might find yourself with a coral reef that is overgrown by an alga that is not native, and you might actually have quite an environmental impact on this. And then you wait 10 years, so you're like way after the crash, and there, I'm sorry, but Mr. Ask is not publishing anymore about, is not communicating anymore about what happens in Tanzania that I could trace. And then you've got the academics left, right? And this is Flower Msuya, who's in Dar es Salaam University, and she tells you that the cotonai, so from the species I was showing, the die-off has had an important impact on the seaweed industry in Zanzibar. Farmers believe that seaweed's properties have changed over time and that seaweed has appeared to lose its weight. So they have also issues with yield that is decreasing. And she's saying that the precarious position many of the farmers now find themselves is exacerbated by the fact that in Zanzibar it's difficult for farmers to negotiate better prices for the crops. So basically they are trapped in sort of a poverty circle because the crop is losing their value. They've got a lot of issues with yield, and they're also in sort of dependency situation with the companies that buy their crops. And the latest paper from Flower was entitled "Seaweed Cultivation in Tanzania is at too high a price to be paid." And really, for me, this is what I would describe a typical trajectory of a new crop, which basically we certainly do not want to reproduce here. I think what we want to have is a sustainable industry, and I think we need to understand what's happened in cases like this to actually mitigate against that. You don't have to look far to actually realize that really history repeats itself. So I've taken here an example. This is the rise and fall of shrimp industry from the 90s. So basically you're just like going back 20 years and looking what's happened in the shrimp industry, and you see it's very, very same story. So in the 90s, shrimp cultivation has been introduced pretty much all around the world, and you have diseases coming in. So one is the Tora syndrome virus, then you've got the white spot syndrome virus, blah, blah, and rapidly devastated the shrimp industry in many parts of the world. And what is important for me here is that both of these diseases were previously unreported. Okay, so, okay. Then another one, the white spot disease, and again a virus. I don't know nothing about viruses. Okay, and they're saying that it was first reported in the early 1990s. By 1994, it had spread throughout Asia. By the end of the 90s, it had spread to the north, the south of Central America, and it is now considered the most serious viral pathogen of shrimp in the world, right? Another one, the yellowhead disease, again a virus, was first reported from mortalities in the early 1990s. So before you start an industry, no one cared about shrimp viruses. That's the bottom line, right? And now continues to be a major pathogen of shrimp worldwide. And then starts another problem, which is that you start looking at the wild reservoirs of these pathogens. And you see here that now that you start looking for them, you find that 18 host species, so 18 wild species of shrimps, can potentially be affected by pathogens. Okay, so the bottom line from here is really that within 10 years of introduction of the industry, you've got four new devastating viruses that have been discovered, okay? And that when you start looking at what these viruses can infect in real life, oh, right, sorry, no. Yeah, sorry. The thing is that, okay. So you have these 4 new devastating diseases discovered. And then at some point, people decided, well, we just have to go breeding for disease-resistant shrimp. And they started the breeding program. And the breeding program was, I would say, a quick fix, right? It was like, you have a pond of shrimps that have all died, except 2, and hopefully there's a male and a female, and you just cross the male and the female shrimp, and you hope that their offspring is going to be resistant. The problem with that is that very, very quickly you end up with a very narrow genetic base, which can be hit even more massively and acutely by the next pathogen coming in. And that's basically what happened, but they started the disease program on the basis of nothing to lose, much to gain, right? And it was basically a race between companies to actually come up with, I would say, the strain that would be disease resistant. And then, sorry, I was getting distracted. We also started to get concerned about natural reservoirs, natural stocks of shrimps and other crustaceans that actually can be affected by these pathogens. And it's very similar to the sea lice in salmon, where effectively the farms can then become a reservoir for pathogens and actually have a very detrimental effect, potentially, on wild populations that surround them. And obviously, there is no research that has ever been conducted on this because no one even knew these pathogens were there within a few years. And you have an industry that is basically unable and ignorant of the impact it potentially has. And there is a huge amount of research that has to be done literally against the clock to try to understand how to minimize and to mitigate the impact. And that's really what we want to avoid with seaweed. Just to finish making that point, I was looking at diseases in the horticultural and agricultural trades, okay? I don't want to be catastrophist, but okay, I was looking at invasive species, right? And this is really looking at the cost of invasive species. So I'm going away from the disease, actually. I'm going more about the cost of introducing non-native organisms in an aquaculture setting, for example, and having this organism then spreading out of control. When you're looking at the cost to society of invasive species, they're telling you that damage and management typically cost hundreds of billions of dollars worldwide. But effectively, the point here, and this is why I've got my laughing call, is that when you look at the 100 topmost disruptive invasive species, they are birds, they are crabs, they are mammals, they are big things, right? There's not a single microbe that has made the cut to being actually a invasive species on this list, right? And really, the bias I want to highlight here is that essentially, mostly because of technical reasons, but that microbes are much less tractable than birds and mammals in the ecosystem, we don't really know the cost of invasive microbes in ecosystems, but we perhaps would be wise to assume that it exists. Then, I like this example about bees, because everyone loves bees, they kind of pollinate our crops and they do honey and it's all great, and they've been introduced everywhere, even in Antarctica, I'm told. And then the thing is that the worldwide bee trade is blamed for dramatic declines of wild insect species, not just bees, it's pollinators in general. And they're, according to this, I would say, pretty serious paper, 20,000 in total of wild insect species that are at threat. And the link is actually, it's not formally established. It's a bit like tobacco, so long you have a bit of doubt, it's not quite established because you've got a massive industry behind it that tells you there is doubt about it. But parasites are suspected. Parasites carried by the bees are suspected to actually be involved or responsible of this decline of 20,000 species worldwide. Okay. Now, if you look about plants, and this is an interesting thing, it's a paper, it's quite old actually, looking at introduction of pathogens in the UK. So we've got a biosecurity framework for plants that is quite strict, okay, in the UK and in the EU. And basically, this journal is counting the number of plant pathogens that have been discovered in the UK over the course of 30 odd years, right? And it's telling you that it's 244 pathogens, so that's a rate of 20 to 30 per year, introduced despite biosecurity being in place, right? And the conclusion of this paper is saying, well, actually not to worry, because you know what? The rate is not increasing, right? So my conclusion is still like, okay, inadvertent introductions still occur at a large scale. And really, this is something we need to also take into account when we start to look at biosecurity in seaweed. And then there is something that has been happening within the, I would say, the early 2000s, and that is a bit posterior to that. It is that, and I think there is this is an excellent paper. It's a massive amount of work. Lots of donkeys involved. You will love it. Effectively, this is a more recent paper looking at the infestation of 3 nurseries with Phytophthora, okay? And they're saying that out of, I think it's 400 nurseries surveyed over 20 years or something like that, 91% of European trees nurseries are contaminated with some Phytophthora pathogen. And at least 49 species out of 69 that they found are non-native, right? And if you look at how they've been discovered, typically they will have been discovered since the early 2000s, also due to improved sequencing technology. And what they're saying is that the trade, the agricultural and horticultural trade, has really evolved from having moved around the world seeds, like, you know, seeds in small bags, to actually moving around flower pots. Because it's now, because of the cost of labor in developing countries and so forth, it's now much more economical to actually grow the seeds in whatever country they come from, and then to import a flower pot with a cubic meter of soil. If it's a tree, you know, you've got a cubic meter of soil that is attached, and that soil comes from that other country, and there is literally no way to know what's in it. And what these people are concluding is that really there should be some evolution of biosecurity regulations to respond to actually the increasingly global trade that we're having. Okay, so now, oh, that's my slide on Eric asking the reaction industry. Okay, so we're again looking at Eric Ask. I've got nothing against him, but I think it's really one of the guys who's actually given an extremely useful testimony across time of the situation as it was perceived in Tanzania. So I think it's a really precious testimony, and you can try to look over time what's happened. So I don't really want to pat him down the wall. That's not the point. The idea is that you look at this trajectory, you know that the guy was, this company that he represents was very involved in setting up the industry in the first place, and you're saying that, you know, it peaked in 2001, crashed in 2008. Okay, and here's what he writes over the time. So from 2003 onwards, he tells you in this paper about quarantine rules for marine animals, and it describes that there are 2 groups of disease, the certifiable and the notifiable. So he knows about biosecurity frameworks in other industry. But it tells you, like, okay, for commercial euchymenids, diseases as such are unknown. Ice, which I was mentioning before, is the only malady that has been noted among the commercial euchymenids. So the irony of it is, if you're a French speaker, malady is the French word for disease. And I was looking at my Oxford dictionary, and malady is some odd English word, malady with a Y, is an odd English word to say disease. So the guy tells you diseases are unknown, but in fact there is one that is known. Look away. Ice is the descriptive name for a malady, so it's not a disease, it's a malady that besets the commercial euchymenids during stress. So they're not diseased, they're just stressed. So, right? So after 30 years of cultivation, no pathogenic agents for the commercial euchymenids have been noted. So he's telling you, like, okay, for all other industry in the world, there are always diseases, but for my industry, it's as such, nothing at all. They are just stressed, and, you know, they've got this weird thing that's called a malady. It does appear, if it does appear during introduction, so they're moving seaweed from different countries, So if you are moving a seaweed from different country and it's stressed by transport, it might get this malady, but it apparently doesn't harm native plants. What data has to back this up? I'm still waiting to see them. During the introduction of Eucuma in Guadeloupe, again, we've not seen the data, ice was rampant, so probably in the introduced plants, but did not spread to neighboring endemic populations. Okay. So basically what I'm saying is that this was written in 2003, you know, when the production was already halved, okay, by someone who was clearly aware of biosecurity elsewhere. And I would say, it seems to me that is completely in denial. So you could say either it's just a personality thing, or either you recognize that industrial stakeholders, and I think this is true, when you look at other examples in other industries, they have a short-term interest to play down ecological issues. And typically this is to avoid regulation coming in. However, what I'm trying to say here is that as scientists and obviously policymakers, the interaction with the industry is key to gather intelligence on changes on the ground and on emerging issues. So the idea is that if you want to design regulation that is necessary, but you want to design it in a clever way, you have to have this interaction with industry. And this is really what I think our job is to promote, and this is what we're trying to do with Global Seaweed Star, is a sort of constructive interaction between all stakeholders to actually maximize the success of the industry, minimize the red tape, but actually be aware that it might have a negative impact, and then really work hard to create a science that actually allows to mitigate it. Okay. So coming back to Eucumate, and I'm just concluding on that, I've not talked about Genial yet. It is really about the situation. When you're looking at this map, what you're looking at is now, okay, we've had local crashes of the industry, okay, and essentially we have all this globalized movement of seed, which Iona is the expert, but basically the regulations are as good as non-existing. And of course, once you've dumped something into the open sea, containment options are extremely limited. So it's not great on land, but it's even worse on sea. So basically, with what I said before, I think we have all the ingredients here for a perfect storm. And really, my 2 pence of wisdom would be that if we're saying this is a global industry that is growing and is now becoming big business, I think Socrates told it first, and then Spider-Man was the last to say this, is that when you realize you've got great power, it comes with great responsibility. And I think that there needs to be that sort of awareness raising. So this is the type of work that we're trying to do as part of Global Seaweed, and then as part of Global Seaweed Star with Les. And basically this is looking here at how crops, so here is my attempt at watching a kelp farm. Here you've got a native kelp stocks and looking at potential impacts of farming kelp. So that could be the sort of escape from the farm of introduced genotypes that would then displace native populations. And then all the business of diseases, so local diseases that would jump onto the crop and actually, you know, lead to a loss, economic loss, but also the crop becoming a reservoir for native pathogens, which would then eradicate the native population that would be sensitive to it. And all the ideas of potentially inadvertently introducing a pathogen when introducing a crop, and that's non-native pathogens, potentially jumping into a local population, and perhaps this invasive crop could be a reservoir for that pathogen as well. So at the moment, the idea is that when you're looking at seaweed, you just have no idea whether that happens or not, and we just do not have the science to actually inform policy. Okay, so coming back to Europe, okay, what is happening in Europe, and what is the status now? Basically, it's been known for a long time, and this is just a paper from Akira, that we have a high prevalence infection, like 70% of kelps, when you're looking for endophytes, are infected, okay? But really, there's been little research done, and you only need to scratch the surface, and that was another PhD student working again with Akira, and they looked at more kelps, and they just found, boom, a new species. And then viruses, other viruses. So this is like within the last 2 years, kelp pathogens are being discovered as we speak. So they're not only discovered, but whether you look at the endophytes or the viruses, I don't know that there would be anyone in the field able to tell you exactly how bad they are, what sort of symptoms they are causing, and whether they're really a problem for the industry or not. So really, we're in the discovery phase. So as part of Genealg, we're doing a monitoring of our pests and SAMS hatchery. So I had to say that, we have a nice-looking seaweed farm. We know how to do nice-looking seaweed, but the ones that we're interested in are the ones that look weird. Okay, and that's Marie-Mathilde's work. And basically, what we want to understand is a baseline of pathogen infestation in the hatchery and the farm, and really try to understand which pathogens are going to be the ones relevant for the industry. I'm not hiding in the data here. It's just we've accumulated samples, but not yet analyzed them. Then we're trying to promote, I would say, a similar community effort. And at the moment, we've not really put much time resources in it, but I'm hoping to increase, to change gear a bit. And I think I've reined in pretty much everyone in the group behind this. And the idea is to actually get extra samples from other people who are trying to cultivate seaweed and to work in a collaborative manner to actually accelerate the disease discovery phase. And the idea is to use this data as far as people allow us to use them openly to produce an online atlas. So this is called My Seaweed Looks Weird. I also sometimes call it Seaweed Anonymous. So I know that some private sector people are a bit worried about the bad publicity of admitting they have diseases in their facilities. So we actually also can handle some data in confidence. Anyway, looking at disease control strategies. So there's been obviously a big research axis over the last few years. So when you're looking at a pathogen coming at the seaweed, you've got different approaches that you can look at. The first one, most direct, is you want to hit the pathogen with something that's going to kill it. The problem of doing this at sea is that this is probably going to be toxic to all sorts of other things, and this is probably the least attractive option and least ecologically friendly. Then you can be looking at making the alga stronger, so somehow giving it some biostimulant, some fertilizer of sorts. Now it's actually more resistant to disease. And there is actually a very nice publication that just came out a week ago, by a Chinese group with Guillain-Barré people looking at stimulating the natural defenses of the kelp with oligogururinates. We've been looking with Swazik Prado at another strategy, which is to effectively try to find inside the kelp microflora some, what we would say, friendly microorganisms who would naturally be there and would actually be able to counteract the pathogen. So it's very similar to Fred's PhD project. But the one I'm going to talk now is the breeding. So really improving the genetic basis of the crop and to breed for disease-resistant varieties. So you've seen we don't have a pathogen yet, but we're starting to breed. Okay. So before you want to breed, you just want to have a bit of a basis for doing it. I don't want to go like the shrimp industry went, just like taking survivors and just hoping for the best. If you want to breed in a proper way, I think you need to have a bit of a conceptual framework. So the first thing we were looking was that, okay, we have different genotypes, algal genotypes that have different responses to pathogen. And then what Martina did, that was a few years ago already, was to look at whether this differences in susceptibility to disease were to some extent heritable. And we've used Ectocampus and Eurychasma as what we thought was an easy experimental system. It's got its own flaws. I'm not going to go into that. But fundamentally, yes, the data we have are consistent with disease resistance being a quantitative, i.e. not a black and white, but a shades of gray character, and it's quantitative, and it's to some extent at least heritable. So we're kind of reassured that if you start breeding, you might get something out of it. And this is where my G-spot moment comes in. It is really where we're now trying, via this GENIAL project, to develop the tools for breeding. And the tool is very much what we call GWAS. So this is Genome-Wide Association Studies. Doesn't mean much, but in fact, it's very cool. Okay. So in order to explain that, I'm going to start with a bit of a digression. Okay. And essentially, I'm just going to look at what is called the domestication trajectory. So the idea is that when you breed, you select from a genetic stock the best performing individuals. So typically you've got like the ancestor of the cow and you end up with a domesticated cow, right? So throughout that trajectory of breeding, you've undergone this process of domestication. This is really the basis of agriculture. It works with cows, it works with all the plants that we grow, okay? Where you have a wild mice here that basically makes very few grains, and then you select for the best performing individuals, and you have a much more viable crop in the end. Okay. And whilst you're doing this, you're effectively having a huge impact, so demographically on your population, genetically, and also phenotypically, so on the outside traits, the characters that your crop expresses. So this is very much the same that is happening for seaweed. And basically here, what is represented on that diagram is what happens when you domesticate seaweed. So the idea is that you've got a population, a species, as this major blob here, this big circle. And the species is made of different populations, which themselves are made of different individuals, right? And populations are all different, and individuals within these populations are all different, and they will evolve over time naturally. And then when you start cultivating, you will typically start from a few individuals that you will then try to propagate and to plant in the field. And this is very much where the Capophycus and Eucuma industry is at the moment, right? It's like 40 years of cultivation, taking stocks from the wild. And then what is happening next is that from this crop, typically, you are going to have better performing individuals, right? So yes, you expand your crop from taking more individuals from the field over 40 years type of thing. And then at some point you realize that some crops really perform better than others. And typically they will be the parents for making crosses and trying to get better performing individuals. So typically you have a sort of funneling down your genetic diversity that is happening. And that is typically where Saccharina japonica would be at the moment in China, where the Chinese have really taken over and over again high performing cultivated plants and crossed them again with each other and essentially narrowed down their genetic basis. All right. And you really have all these sort of genetic bottlenecks, right? And now again, I'm like going a bit tangentially to what you can do with genetics as a researcher. And really this is what I call the holy grail of physiology and functional genomics, is to understand what genes are doing. And genetics is a way to actually do this. So say that you're interested in trying to understand what gene is behind disease resistance. You have what is called the classical genetics approach, which you probably have all heard about in high school with Mendel and green peas being wrinkled or not wrinkled, with Drosophila with curved wings or not. So typically what you have is that you have a wild type that has a phenotype, in my case, say, resistant to disease, and you find a mutant which has the opposite phenotype. And then basically, the genetics approach is that you cross this wild type with the mutant, and then you build a genetic map, and you map the gene, and it's a long amount of work. And here, basically, what I'm trying to say, this is a unit introduced by Ceci. It's called like the donkey. And it's a unit of dull and repetitive work. And basically, finding the mutant is typically finding your needle in a haystack and making the crosses and then mapping back on your genetic map the gene that is responsible for the phenotype that you observed as a huge amount of work. Early mid-90s, early 21st century, comes the first genomes, and with that, a complete different approach that is called reverse genetics. And effectively, reverse genetics is based on the availability of genomes plus transgenesis, right? And this is what got scientists excited about GMOs. So the idea is that the starting point is the same. So you've got a wild type that is resistant to disease, but the genome information also gives you a candidate gene that maybe that you hypothesize is responsible for the phenotype. Then using transgenesis, you just like knock out that gene. So essentially you create the mutant. Instead of looking for it, you just create it, boom, right? And then you look that this mutant has lost the wild type phenotype, and then you conclude that this gene Y is necessary to confer disease resistance. And then the second part of the demonstration is actually to put back gene Y in the mutant, right, and to restore the original wild type phenotype, which tells you that this gene Y is sufficient to confer disease resistance. So it's necessary and sufficient, so it's responsible, and that's really the very elegant, straightforward approach, no donkey work anymore. Problem we have with brown algae is that we do not have a way to disrupt genes. So we're working on it as well, and that's what Yassine is doing in the Simon Plus in Roscoff. At the moment, that is not an option for us. Okay. Another layer of complexity is that not everything in life is black and white. So typically, then you have to introduce what is called quantitative genetics, and it's the very same principle, but instead of having like a wild type that is resistant and a mutant that is susceptible, you have a wild type that is somewhat resistant to disease and a mutant that is somewhat susceptible. So there is a difference, but that difference is subtle, right? And basically, this is the very, very same approach, but then you can only find that one gene, that is also called a locus, that's where you've got loci here, confers resistance to disease to some extent. So this is the same amount of donkey work, except that you also need to have the stats. So your donkeys need to know their stats, right? Okay, and at that point, you're like, you're hating genetics. And here's now GWAS coming for you. And there is some beauty about GWAS, which is to say, why would you bother to do all these crosses and all this work when in fact nature has already done it all for you, right? So the idea is that if you remember my problem with my species, you have lots of different populations with individuals that are all somewhat different, right? So why not take them? Okay. And then take as many of them, biobank as much of that diversity as possible to increase the chance of gathering all the alleles that are going to be responsible for the best phenotypes, okay? And let's put them in a culture collection, right? So that they will be available for posterity. And then you genotype all these individuals, and then you phenotype all these individuals for your trait of interest, for example, disease resistance, but it could be growth, it could be yield, protein content, whatever you want. Then do the stats. So I mean, correlate the genotypes with the phenotypes, and then find the genes or the QTLs, whatever you want to call them. So find the genes responsible for trait of interest, and essentially backpedal to, okay, I know this gene is involved in that trait of interest. So somewhere on that chromosome, I know I've got a gene that is responsible for that trait, or 15% of that trait, and then I can backpedal to the best performing individual had that specific form of that gene, that specific allele, and that could be my parent for a future breeding program. So effectively, once you have this database built, you can backpedal and identify back in the natural population who is carrying the best alleles and who you should be crossing to actually get your breeding program, right? And it means that if you have here this red population that comes from this yellow, so I'm coming back to the cultivating population of Saccharina japonica was red, you can actually identify that in the blue population, you've got a subset of individuals that carry a very interesting gene and that you would want to actually start breeding with them, right? So, donkey work. You have a lot of individuals. You all want to biobank as much genetic diversity as possible. So you see the donkeys appearing here. Genotype all individual, phenotype them, do the genotype and phenotype. It's the same as a QTL approach, right? So basically, it's a lot of work, and it involves a lot of stats. But the beauty of it is that it's a one-off, okay, hopefully. And once you have it in a culture collection, it's freely accessible. And then whatever you're interested in afterwards, whether it's protein content or whatever, is you just find a way to measure it and to correlate your phenotype with the genotype that's already in the database. And that, once you've made this one-off investment, which is genealc, then the vision, hopefully, is that anyone could come and measure any trait they're interested in. Just to show that it does work, this is actually just randomly taken from, I've just Googled it up yesterday, I found it funny. So it's wheat breeding at the moment. So this is wheat, right? And they're looking at making bread, and the trait that they're interested in measuring, so really whatever you want to measure, is effectively the resistance of the dough, right? So this is a machine that blows air into your dough, and until it bursts. It's a bit like a bubble gum with bread or dough, okay? And then when they were looking at something that they call, so this is called an alveograph, I think. And then they were looking at the loaf volume, and they were also looking at the size of the bubbles inside the bread once it's baked, I think. And they measure it, so they just found a machine that can measure this parameter, and then they do the stats, blah, blah, and then they find that, yes, so on these chromosomes here, you've got like specific places, specific genes that are involved in that trait. And then they can go back to the parents that actually perform better in that test and gives you the best bread. Okay. It is routine, so it works, but it's not just like stretch of mind by scientists. It actually is routine now in plant and animal breeding. So I just like, again, Googled up, it was very, got hundreds of papers like this. So you've got a lot of genome breeding, for example, on dairy cows, every vegetable you can think of. And what I want to actually highlight is that Yassine, our own very own Yassine, has done a PhD on this, on alfalfa and the pathogen, and that was actually a deciding reason to get that skill set here at SAMS when we hired him. And I think it's the usual story, right? Getting over enthusiastic. Essentially, we've started with brown algae, looking at QTL mapping. I'm going to say we, that's more the Roscoff side that is leading Genealg. And essentially, they've last year published a paper showing that, yes, when you look at Ectocampus as a model brown alga, and you take a dad and a mum that are somewhat have a somewhat different response to different temperatures, they can find loci, so places on the chromosomes that are involved in resistance to temperature, resistance to salinity. So this is the first example I'm aware of on brown algae. Not entirely sure it's got applications, but it's a proof of concept. Now, yes, it does work. And essentially, what we're doing at the moment is now trying to find ways to measure disease resistance in Ectocampus. And the idea is really to look at getting numbers, and this is Pedro doing this. It's really to look at non-invasive continuous monitoring of growth, of pathogen infestation, and also having some endpoint measurements of how much pathogen we have in a disease assay. So essentially, we are now working on our phenotyping to get the numbers we need to match with the phenotype. I think this is no, I've got another 5 slides. Then the biobanking step, I think is the one that has been a massive effort, and it's been led by Marie-Mathilde. So it's really, that step is to biobank as much genetic diversity as possible to increase the chances of gathering the best alleles for a breeding program. So the idea is that you can see the donkeys accumulating because you can sample all Saccharina all across Europe, so many you can. And the idea has been really for Marie to kind of try and design a sampling program that maximizes the genetic diversity we have, whilst minimizing the number of individuals we have to look after in the culture collection. So like, it involves a lot of population genetics that I'm not going to get into, and hopefully by having a clever sampling program, we can remove a bit of the donkey work. Okay, and that's where we've been. So we've been from Portugal, Spain, a lot in France. We're trying to get a lot of the United Kingdom, Norway, Denmark, Netherlands, and so forth. There was plan in Canada, but I think no, Canada is not going ahead. We're trying to cryopreserve them to, again, limit the donkey work of maintaining them. And then this is, I think, my last slide. Once we have all this biobanking in place, which at the moment is ongoing, the idea is to genotype all individuals. I'm not too sure how we're going to do that, but it's been done before. Alex Thompson here in his PhD is doing it, and Roscoff also has the technology. And then we want to phenotype all the individuals. Okay, so the phenotyping assays that we're just talking about, we have essentially looking at working with plate readers to actually try to make it a bit more streamlined. And then once the assays are working, which is eminent, we want to phenotype all the individuals, and that's where really another bunch of donkeys are involved. So we've got our two chief donkeys, Maria and Ceci. We've just been recruiting, so we're still hiring. We've been recruiting a new chap, Callum, who's going to start on the 1st of April. He's been warned and he's signed. And if the resource committee rubber stamps it, there should be another 2 positions coming up. And not just to do all that, just that, because you get crazy if you do that full time, but really to contribute to that effort as well. And that's kind of it. Thank you very much. I don't know if there is time for questions. I'm sorry for being so long. Yeah, Joseph.
Okay, so it's great. I have a lot of questions, but one of the beauties of parasites is that they are always entangled in this co-evolutionary arms race with their host. So, for instance, you have a lot of experiments here trying to select for the best phenotypes and what on the host side.
Mm-hmm.
But what assurances you have is that after all that work, when you're characterizing the best genotype, the best crossbreeding, there's not a parasite there that will just co-evolve and target that particular mutant? Because I presume that in the lab here you only have several parasites that you're working with, or maybe not. I guess my point is that they may not be the most abundant one, but only reflect the places where they were isolated, associated with the host.
No, I think it's a perfectly valid question. I would say there are several layers of answers to that. I would say the most immediate answer is you've got to start somewhere. And I think at the moment, we just have no idea of the genetic basis for disease resistance. I won't say no idea. I think we've started to work out by indirect ways. And I think you're right. I think there are the 2 approaches. What we're doing in the lab is with a number of clonal strains and so forth. So we are hitting that limitation. But when the beauty of GWAS is that you're looking at natural populations that have been evolving for millions of years, or so much time you think, under all these selective pressures, right? And basically, you should have the diversity and the polymorphism in these populations that would allow you to respond to, say, a virus or an oomycete or whatever. And then there is also the complexity of trade-offs between resistance to different pathogens. So this is something that, at the moment, we just do not have any idea whether it exists on brown algae, but I think there is an example that comes to mind for me. There is a gene in Arabidopsis that is really one allele of it confers resistance to an oomycete, the other allele confers resistance to a virus. So it's the same gene that has actually been identified by different groups that were working on different things, on the 2 different pathogens. And they just, once they did the genetic mapping, like they spent years and years and years of genetic mapping, and to realize that they were hitting the same gene, but two different alleles. And we just have no idea of that. But I think it's 20 years down the line that we'll be able to address this question.
That leads me to, another question that's related to that. Cost associated with resistance. So you have obviously identified some of these resistant phenotypes. What are the potential costs of the slower growth rate?
We have no idea about that yet. We have no idea about that, but part of the phenotyping assay that Pedro is setting up at the moment is just like and I think we're the first people able to do this, is to build a non-invasive growth curve of our alga. So it's really, we're starting from scratch here, and I think we're the first ones being able to actually measure the growth of a macroalga non-invasively in a multiplate reader. And that means that we're going to be able to start asking that type of questions, but we've not started. I think we're really starting from low. Other questions? Everyone's hungry? Okay, then. Thank you very much.